खाना खाना, मैं नहीं एक ज़रूरी बात तो रह गई भी, मैं पहले बात कर लूँ। तो वो देखो ना, 10 सवा 10 से हमने बुलाया तो कहीं नहीं, ऑनलाइन ही होना है, या जैसे जहाँ पे बच्चे होंगे वहाँ पे हम सभी होंगे। तो 10 सवा 10 बजे मतलब कि मुझे ये है कि चलो मैं कपड़े डाल के आ जाऊँगी फटाफट से 9 सवा 9, या तो फिर मैं हॉस्टल आ जाऊँगी आपकी तरफ की, है ना। मेरा एक बार फॉर्म वगैरह कर देना, बस अकेले बालों का ही थोड़ा सा मुझे है, और तो हाँ। हाँ, मैं तो कुर्ता ही डालाऊंगी अपना, वो सर्दियों वाला, या ब्लैक डाल लूँगी या दूसरा पिंक डाल लूँगी। हाँ, वो वाला ब्लैक वाला नहीं है जो मैं डाल के तो आती हूँ यार रोज, एक दो ही तो है कुर्ते। हाँ, एक ब्लैक है। हाँ हाँ हाँ, ब्लैक जिसके से आगे के फूल से लगे हुए हैं, वो ब्लैक ही है। जिसमें जिस कल जो डाला था, जिसमें मैंने आपको बताया कि मुझे बस वो चाहिए, सेफ्टी पिन चाहिए कि उसका गला बड़ा है, परसों डाला था। हाँ, मैंने कहा काम कर दोगे और वो लिपस्टिक लगा दोगे। हाँ, या कोई मेरे पास जो है वो भी मैं उठा लाऊंगी, कि चलो थोड़ा सा देख के फटाफट से रेडी हो जाऊंगी। चलो ठीक है, बस फिर मैं आपसे आके मिलूंगी। तो अब हाँ, अभी मैं कर लूँ डिनर, फिर मैं बात करती हूँ आपसे। चलो ओके जी, आपने खाना खा लिया? नहीं यार, भूख लगी, खा ली थोड़ा बहुत। चलो दूध पी लेना, दूध पी लोगे ना? चलो ठीक है, फिर मैं आपसे बाद में करती हूँ। ओके। So yes, good evening to all. My voice is audible. Let me share the link in the group once a time. So are you able to get me? My voice is audible. All of you, please let me know my voice is audible. Let me share the link. Good evening to all. My voice is audible to you. Yes, good evening to all. My voice is audible. Good evening to all. My voice is audible. Hello. Okay, okay, okay. Thank you, thank you. Shubham, Yaman, thank you a lot. So yes, today we are meeting little bit early because I have to take 2 hours long session. That is why I am little bit early here. So yes, good evening to all of you. I am just waiting for all of you so that we will start the session. And we have to discuss lots of things today. How is your day today? Everything is fine? Everything is fine. So how is your Sunday today? You all are free or you have your exams or something? I want to take it a little bit early because then I came to know that many of you have some exams or some extra, work for that. So I decide I will take it in the night only. So how are you all? Everything is fine. My voice is okay. Are you getting me properly? Yes, yes. Good evening, Simran, Mugdha, Shruti, Monojit, Harshani, Mugdha, Pritam. So good evening to all. Good evening. Okay, okay. Shruti, good evening. Okay. Yes, I am good. I am fine. Yes, today let me share the syllabus. Let me share the topics which we have to discuss. If the session will go smoothly, that means there will be no network issue, then I will try to take your session a little bit longer, and immediately after the session I will share the PPTs with all of you. Okay. So we are just waiting for the students to join. Otherwise, if anyone will not be able to join right now because of some their own schedules, so we will share the link with them and they can watch it at any time. When he or she will be free, they will watch it at any time. So no issues at all, because I have to start the session. I have to complete the topic today. So yes, I have to start it. After, at the end, definitely at the end I will answer your queries, and at the end we will discuss about review papers also. But now I will start with the session. I will start with the lecture which I have to deliver. So this is the syllabus which we all have. We have already discussed cell culture techniques. We have discussed basic principles. Now today I will start with recombinant technologies. Okay, recombinant technologies, and also we have to cover molecular biological techniques, the techniques which are there in the MBBT. So we will try to start with recombinant DNA technology, and then we will move further to the molecular biology techniques which are there, the techniques which are basically used in the field of molecular biology. So I will try to do these things. I will try to complete those topics which are little bit easy, and it will be quite interesting for all of you also, because some of you are totally new to it. So I will try to start from very basic. Okay, so I will try to start from very basic, and I have to cover the topics which are related to recombinant DNA technology, and some of the molecular biology techniques. So yes, Anvesha, Mahesh, Chandrala, Anukriti, Mitrika Goel, good evening to all of you. So I will start today recombinant DNA technologies. If you have any questions, I will handle it at the end of the presentation, or even you can message me at any time. I will definitely answer your queries, no issues at all. But yes, I will try to cover some important topics today. So I hope all of you will cooperate, and we will discuss anything regarding the review paper also at the end of the class. Divya Jyoti, it's not good afternoon right here. I'm sorry. Maybe you are from, like, one of the student in our class is from France, from Italy. So maybe you are also from the outside. So here we have nights only, but still it's okay. So good evening to you. Okay, okay. Good evening. Good evening. Because I think one student is there. He or maybe she is from Italy. Yeah, you are from Italy. Okay, okay. So yes, most welcome to you, and I am quite surprised that you are listening to my sessions right here from Italy. I want to know what are the study you are pursuing in Italy. If you are comfortable, please share. No issues, Elvia. The topics which are left, I will let you know. No issues at all. Don't worry. So if you are comfortable, Divyo Jyoti, please let me know which study or what are you pursuing in Italy right now. We have Sudarshana Gupta. Good evening, ma'am. Good evening, Sudarshana. Yes, from tomorrow onwards, I will try to keep my camera on also, so that you will be able to see me at least. Okay, so she is pursuing MS in molecular biology and genetics. Okay, very good, very good. MS in molecular biology and genetics. So you must aware of all those things which we are discussing right here. You must be aware of all these things, the techniques which are there in MBBT. I hope so. Shivam, hello, how are you, Shivam? Okay, so I'm now just going to start the session. Yes, Neha, please ask. So we are just waiting for the students to join. Then we will start the session. So we have Shah. Shah, give it. Good evening. How are you? Yeah, Shivam, just give me some time. After this lecture, I will be free and I will go to check all those abstracts and the topics, and then I will definitely mail you back if there is any type of error. I will let you know, Shivam, no worries. Yeah, Dhruv, I already, sorry, it's not Dhruv, it's Dhruvin. So I already told all of you that we will discuss everything regarding the review at the end of the session, okay? So we will discuss everything regarding review, how to write it, how many words it should be, how many pages it should be, every query regarding the review we will discuss, but after the end of the session, because otherwise we will not be able to complete our lecture within the 20 days. Okay. So I know all of you have many queries. We will discuss it definitely. So Neha, you are asking something. I am just waiting for your question. Ma'am, can you tell me one thing and then you are not asking me. Okay, so then I am now going to start the session. We have to start the very first topic, which is recombinant technology, and I will start from very basic, definitely. So yes, Shafaq Hasnan. Hi, how are you, Shafaq? Okay, so now I am going to start the session. I will answer all of your queries after the end of the video. So today we have to discuss the topic which is recombinant technology. Okay. So I know many of you have done this before also, but this is an important part of the MBBT lecture series. So I will take a little bit extra time to let you know about everything regarding the recombinant technology or recombinant DNA technology. So in this we have to cover the introduction, the discovery, the goals, objectives, what is the procedure of recombinant DNA technology, the enzymes, the vectors, the techniques, everything we have to discuss in detail. Now what is basically recombinant DNA technology is? So what happened here? DNA molecules that are extracted from different sources. So DNA molecules, you must know about something about the DNA first. Like there is where the DNA is present basically. I have to ask this question. So where the DNA is present basically, the DNA or the RNA or the genetic material, where it is present in the cell. Any of you please let me know what is basically DNA. DNA is a nucleic acid. It is a genetic material, na? So where it is present inside the cell? All of you. Yes, all of you. So DNA is present here basically. And from if there is DNA and RNA, if there is DNA and RNA, which which of the genetic material or which of the nucleic acid will move outside from the nucleus. Yes, the DNA is present in the chromosome or inside the nucleus, but out from both DNA and RNA, which nucleic acid will be come outside. That means from the nucleus into the cytoplasm, which nucleic acid will be come outside. Yeah. So DNA is the genetic material. DNA will remain inside the nucleus always, and the message will be received by the RNA in the form of messenger RNA, and that RNA will move out from the nucleus into the cytoplasm. Okay. So this is the yes. Okay, okay. So I just let you know that there is some techniques like DNA isolation and RNA isolation. In MBBT, in molecular biology, we have to do, if we want to work on the DNA, so we have to isolate that particular DNA from the nucleus, okay, or from that particular group of cells. So there are some procedures which are known as DNA isolation or RNA isolation. We have to isolate that particular DNA from the cell, and later on, after homogenization, after using the lysine buffer and using the whole protocol, we are able to isolate the DNA at the end of the reaction, and that DNA will be run on either on the that DNA will be run on agrocell electrophoresis if you want to pursue it further for the PCR or for your any further applications. We have to isolate the DNA and RNA. So there are two terms, there is DNA isolation and there is RNA isolation. So there are two terms, DNA isolation and RNA isolation. Both DNA and RNA are isolated from the cell during this molecular biology technique, or if we want to do, particularly want to do work on DNA. Now DNA is present inside the cell, okay, you all know it that DNA is present inside the cell, but there is one more DNA which is known as mitochondrial DNA. You have the DNA which is inside the nucleus, which is known as nuclear DNA. But there is another DNA which is present inside the mitochondria. Mitochondria is the organelle of a cell. Mitochondria is semi-autonomous in nature. Why mitochondria is known as semi-autonomous in nature? Any one of you? Mitochondria is the organelle which is present inside the cell. Mitochondria is known as semi-autonomous in nature. What is the meaning of it? Have you any knowledge about the mitochondrial DNA? Any of you? The DNA which is present inside the mitochondria? Like we have the nuclear DNA which is present inside the nucleus, which controls the whole activity of the cell. Whole activity of the cell. So that is controlled by the nuclear DNA. Similarly, we have a mitochondria. Yes. Moving mitochondria is known as energy house of the cell, and it has its own DNA which is present inside the mitochondria. So that is why it is known as semi-autonomous in nature, because mitochondria is not dependent on the nuclear DNA for its functioning. So that is why it is known as the powerhouse of the cell, or you can say energy house of the cell, and it controls, yes, it is a source of its own activity, so it doesn't depend upon the nuclear DNA. So we have two types of DNA which is present inside the cell. So when we are working on the this molecular biology, when we want to isolate, we have two type of isolation. Either we are able to get the DNA or DNA isolation, which is the genetic material, and even some workers or some researchers works on the RNA. So for that, he or she needed the RNA isolation. So this is the only the basic thing which I want to let you know before starting the recombinant DNA technology. So from the name implicates, in the recombinant DNA technology, we are able to extract the DNA. So you must know that DNA extraction is there. DNA extraction is a procedure when we are able to isolate the DNA, isolate the genetic material from the cell. I will discuss that isolation with all of you, maybe in this PPT or in the next PPT. So, DNA molecules are extracted from different sources, or you can say the DNA are isolated from the different groups of cells and chemically joined together. For example, DNA comprising an animal gene may be recombined with the DNA from a bacteria. So we have animal cells. So this is a cell. In the cell we have a nucleus. In the nucleus we have a DNA, which is a genetic material. So what happens here? We just isolate that particular DNA or that particular segment of the DNA, and then we recombine that particular segment with the DNA from the bacteria. So it is basically recombinant DNA technology is the technique, or it is a technology when we are isolating the DNA from different sources. We are isolating the DNA from different sources, and then by some method we are able to join them together. By some method we are able to join them together. Getting my point? So for example is that we are extracting the DNA from the animal cell and we are just joining them with the DNA from the bacteria. So this is basically recombinant DNA technology. We are isolated the DNA from different sources and then later we are combining them together. So now this is the discovery of recombinant DNA technology. How this technology comes into existence? How? So we all know that it was Watson and Crick in 1953 who discovered the structure of DNA. They told us that DNA is double helical or coiled structure, helical structure. According to Watson and Crick, it's a double helical structure. In the year 1953, we are able to know that it consists of sugar base, or you can say pentose sugar, along with the nitrogenous bases and along with the phosphate group attached in case of DNA. So we are able to completely know the structure of our DNA in the year 1953. Then in 1967, we are able to isolate DNA ligase. So from the name it implicates ligase is the enzyme. So in the year 1967, we are able to isolate the enzyme which is DNA ligase. In the year 1970, we able to isolate another enzyme which is REase, or you can say this enzyme was isolated specifically for the RNA. In the year 1972, this was a year when Paul Berg, the scientist, was able to generate the term which is known as rDNA technology in the year 1972. So DNA comes into existence by 1953. We are able to join those DNA segments together in the year 1972. Then again one scientist, Cohen and Bohr, the scientists are there, which are able to produce first plasmid vector capable of being replicated within a bacterial host. So we are able to get the plasmid vector also. So this is the brief history of the recombinant DNA technology, how it comes into existence. Now, what is the need of these things? What is the need of recombinant DNA technology? What is the need of combining different genes together? Why we have to combine? First, we have to isolate the DNA and then we have to combine it with another DNA. Why? So, these are some important goals of this technology: to isolate and characterize a gene, to make desired alterations in one or more isolated genes, to return altered genes to the living cells, artificially synthesize the new genes, alternating the genome of an organism, understanding the hereditary disease. So this is the most important thing, that by using rDNA we are able to control some hereditary disease. We are able to cure some hereditary disease. We are able to synthesize new gene. We are able to alter, or we are able to com completely change the whole genome of an organism. We are able to improve the human genome. We are able to isolate and characterize a gene. So basically, we are able to particularly work on a particular gene. Suppose we want to work on, like in mitochondrial DNA, we have CO1 gene, CO2 gene, 16S gene, 12S gene. So you are able to isolate the DNA, and from that particular DNA you are able to work on a particular group of gene. So these are the functions of RDNA technology. Now what is the basic principles which are there in the RDNA technology? The number one thing is generation of the DNA fragment and selection of desired piece of DNA. Suppose this is the DNA segment which you isolate from the cell. But now on which particular segment you want to work on, which particular segment you want to carry further. So generation of the DNA fragment and selection of the desired piece of DNA. Insertion of the selected DNA into the cloning vector. Suppose I want to take that particular DNA sequence, then I have to take that sequence and I have to add this particular sequence into the cloning vector. I will show you the diagram also. Okay, so insertion of the selected DNA into the cloning vector to create rDNA or chimeric DNA. So this particular segment, suppose I want to take only that particular segment, only that. So I have to cut this particular segment and I will insert it in a cloning vector to form the rDNA or chimeric DNA. Introduction of the recombinant vector into the host cell. Then this recombinant vector or a cloning vector will be introduced into the host cell. I will show you in the diagrams also. Then multiplication and selection of the clones containing the recombinant molecules, expression of the gene to produce the desired product. So all these things are there in the case of rDNA technology. But the very first thing which we have to do is we have to get the DNA. From that DNA we have to get our own particular sequence on which we want to work. On that particular sequence we have to add it in a cloning vector. That cloning vector or the recombinant vector will enter into the host cell. So these are the procedure of making the rDNA. You have to keep in mind. The very first point is isolation of the DNA. Then cutting off the particular DNA segment on which you want to work. Then third is joining of DNA, and fourth is amplifying of the DNA. So number one is isolating a DNA. As I told you before, DNA isolation or DNA extraction is there, from which we are able to get the DNA, the genomic DNA. So we have to lyse the cell, we have to use lysine buffer, we have to use homogenizing buffer, centrifugation is there, and at the end what we will get the DNA of our point of interest. So suppose this is the group, this is a cell, and the nucleus, DNA is present inside the nucleus. So after DNA extraction and digestion, we are able to get the desired DNA on which we want to work. So these are the DNA on which we want to work, DNA fragments on which we want to work. So this is the number one step. Are you getting it or not? Please check, let me check it. So everything is fine till now? Are you able to understand what is rDNA? Are you able to get it? Everything is fine till now? So that I will keep going. I am just going to check it so that I came to know whether you are getting it or not. So everything is fine till now? Please let me know. So the very first step which is there, it is the DNA extraction. So you have to remember all these points. So we have to start with the very first step. The very first step is the DNA isolation or DNA extraction. From the nucleus of the cell, we have to take the DNA, and from that DNA, we have to take the particular DNA fragment on which we want to work. So we have to take the this one, we have to take the particular DNA fragment on which we want to work. So this is the first step. This is the first step in case of our DNA technology. So you have to keep in mind the very first step is the isolation of the DNA. So I hope I must give you some homework yesterday also, maybe not, but I will give you the homework from these PPTs today. So please try to get it properly. So the very first step is the isolation of the DNA or DNA extraction. Now number two step is the cutting of the DNA, because DNA is a large molecule. You want to take a specific sequence or you want to take a specific fragment only. So DNA can be cut into large fragments by mechanical shearing or by the use of restriction enzymes. So we have restriction enzymes, which are basically the enzymes which will cut the DNA at the proper size which we want, or the proper fragments which we want. So these are the restriction enzymes. Suppose this is the DNA fragment and you want to take the specific sequence only. So what are the restriction enzymes do? They will cut at the desired end on which you want to cut it. So a special class of sequence specific enzymes, they are found in bacteria. Site specific leave DNA molecules only at a specific nucleotide sequence. So these enzymes, these restriction enzymes recognize the DNA based sequence that are palindrome. If you are able to see it, now you have to tell me what are the palindromic sequences. I hope all of you must know it. What are the palindromic sequences? So these restriction enzymes make staggered cuts with complementary base sequences for easy circulation. So let me show you in this diagram. So we have the specific enzymes which are the restriction enzymes. So this is the DNA segment from 5 prime to 3 prime, and here it is complementary to it, 5 prime to 3 prime. So suppose I want to cut the sequence on that end. So these enzymes will cut the sequence. Okay, these are the enzymes. Eco R1. So these are the restriction enzymes. So this is the DNA 1, or you can say one DNA molecule. This is another DNA molecule. Now what will happen? So they cut it here. Now this is the one DNA molecule, and here it is cut here. If you are able to see this thing will be deleted. Only this thing is here. CG. So both of these sequences will merge now. If you are able to get it, this is the dotted DNA. That means this is the DNA molecule 1. This dotted is the DNA molecule 1, and this is the DNA molecule 2. So with the help of restriction enzymes, we are able to merge two DNA molecules together. So if you are able to see here, this is the different DNA molecule. This is the DNA molecule A, this one, and this is the DNA molecule B. So by using restriction enzymes, we are able to get the desired sequence which we want to work on. So with the help of DNA ligase, the very first enzyme which was isolated in the history, so with the help of DNA ligase, we are able to join that two DNA fragments. Getting it? So with the help of the enzyme which is DNA ligase, we are able to join two DNA sequences. It may be from the humans, it may be from the animal tissue, it may be from the bacteria, but we are able to join these two sequences with the help of DNA ligase. But DNA ligase will work only when the enzyme will break it into two sequences. So this is the thing, cells are here, they are extracted and digested. We are able to get the desired DNA fragments. Now these DNA fragments, they are inserted inside the vector. And from here they are inserted inside the bacterial cells. So these cloning vectors which contain the DNA from these cells, they are inserted inside the bacterial cells. So if you are able to see, this red one is here, the green one is here, and the purple one is here. So these segments from these DNA, from these cells, we just inserted in the bacterial cell. Getting my point? So this is the whole thing how we are discussing about the rDNA technology. So I just want to give you know about, I just want to tell you about the basics which are there in the rDNA technology. So if you are able to see here, this is post-human chromosome. This is only the representation only, diagrammatic representation. So suppose I just want to take insulin gene. Suppose we have so many long sequences or the human chromosome is there. So with the help of restriction enzyme, what I do, I just take the insulin gene. Getting my point? The very first step is the isolation of the DNA. So I just take the particular gene on which I want to work. So with the help of restriction enzymes, they cleave the insulin gene from the chromosome. So the enzymes will break and we are able to get the desired gene which is in the red color. So restriction enzymes cleave into chromosome and create corresponding gap in the plasmid. So this red gene which we want to work on, this will then get inserted into the bacterial plasmid. This one. So this gene get inserted into the cloning vector, or you can say in the bacterial plasmid. So this red one get inserted in the bacterial plasmid and with the help of an enzyme DNA ligase. So here we have the DNA ligase enzyme which will which will join the two DNA molecules. So this sequence, now this DNA sequence along with our desired sequence which is in red color, it gets entered into the bacterial cell. Okay. So the transfer plasmid is inserted into the bacterial cell. So this plasmid is entered into the bacterial cell. Getting my point? Like here, this plasmid which is joined with the help of DNA ligase, it will enter into the bacterial cell. So now it enters into the bacterial cell. What will happen now? Now it will divide and it will grow. So after mitosis creates many bacterial clones containing the insulin gene. So because we all know that this is the property of the bacterial cell, that it will do the mitosis and it will able to replicate its own self. So we are able to get that much of the insulin gene. So insulin gene inserted into the plasmid export insulin which can be collected from the human for further use. So we will able to get so many copies of that particular gene, a single copy. But by whole this process, we are able to get so many copies of the insulin gene. Now why we need so many copies of an insulin gene? Suppose if we want to work on diabetes, some researchers are there who are working on the diabetes or for any another functioning of the body, we need the insulin gene so that we will be able to know its functioning in detail. So this is the basically the whole procedure in case of the recombinant DNA technology. First we have to select the particular gene on which we want to work. Then with the help of enzymes we have to take that particular gene. This particular gene is then entered into the bacterial plasmid with the help of DNA ligase. So with the help of DNA ligase, this particular gene is entered. After it will be entered into the bacterial cell, because the bacterial cell has the capacity or the ability to grow, to divide, to replicate itself. So it will be replicate here. So by this way we are able to get the insulin gene as much as we need it. So this is the main thing this is the whole thing which is there in the rDNA. If you are able to understand it, then you will be able to understand the whole technology. So up to now, is it clear to all of you? Let me check it. Yeah, you are right. The same repeated sequences are known as the palindromic sequences. Yes, very good. So up to now, is it clear to all of you? so that we will keep moving. Please let me know. Yes, I will let you know about the DNA extraction. The DNA extraction will be discussed in some another topic, but I will discuss it definitely. So shall we continue? Everything is fine right now? So this is the whole procedure of the recombinant DNA technology. Because some students are totally new to it, that is why I am just starting it from very clearly. Okay, Preetham, no issues. I think after the end of the presentation, your doubts will be clear. No issues at all, because this is very new thing. Some of you are totally new to it, so you may have some problem. But let me complete the PPT. I hope all the your doubts will be cleared then. Okay. So, but this is you have to keep these steps in mind. You have to remind all these steps that we have to isolate the DNA. Number one. Then after the isolation of the DNA, from that DNA we have to take the particular fragment on which we want to work. Number two. Okay. After it, what happens? That particular DNA fragment is inserted into the vector with the help of DNA ligase enzyme. That particular plasmid or a vector, it get enters into the bacterial cell. Now why we have to enter it into the bacterial cell? Because bacterial cell has the ability to replicate as much as we want to. So that is why this is the whole process of the biology. Okay. So now let me complete it. So now we have to discuss what are basically restriction endonuclease. I already told you this diagrammatic version of RDNA technology. Now what are restriction endonucleases? What are restriction endonucleases? We already discussed it before that they are the enzymes which are needed for the manipulation of the DNA. They are the enzymes that can cut or split the DNA at the specific site. So this is the DNA, the very long fragment, but we want to cut it here. We just want to take that particular segment. So this is for the insulin gene, or this is for the any another gene, or the CO1 gene or the CO2 gene. So these enzymes will cut it on a particular or a specific site. These were first discovered in E. coli, restricting the replication of bacteriophage by cutting the viral DNA. So thus the enzymes that restrict the viral replication are known as restriction enzymes or restriction endonucleases. So they are also known as restriction enzymes or restriction endonucleases. And what is the function of these enzymes? They have the ability to cleave the DNA molecule. They just cut the DNA molecule. They are also known as the scissors. That is, they are cutting the DNA molecules. Now we have another term which is recombination sequences. So recombination sequences is the site where the DNA is cut by the restriction endonucleases. So this is the site where the DNA cut it. These are known as, these sequences are known as recombination sequences. Restriction endonucleases can specifically recognize the DNA with a particular sequence of 4 to 8 nucleotides and then cleave it. After it, we have the cleavage pattern. Now what happens after the cleavage? When you cut the sequence, after it what we will have? We have to, by the use of DNA ligase, we have to join it with another DNA fragment. So what is the cleavage pattern? The DNA fragment by restriction endonuclease may have mostly sticky ends or blunt ends. I will show you the diagram. So DNA fragments with the sticking ends are particularly useful for rDNA experiments. So we have two types of cleavage. One we have sticky ends and another is the blunt ends. Now this is your homework. You have to tell me what is the difference between sticky ends and the blunt ends? Even it will be discussed in the PPT also, but till it's your homework, what is the difference between sticky ends and the blunt ends? So DNA fragments with the sticky ends are particularly useful for rDNA experiment, since single-stranded sticky DNA ends can easily pair with any other DNA fragment having complementary sequences. We have two types of sequences. We have two types of cleavages. Let me show you. Yeah, we have sticky ends. So these are the sticky ends when we are cleaving it in between, and we have the blunt ends. You have to tell me the difference between those two types of cleavage and which cleavage is more prominent. So this is your homework. Okay, please keep it in mind. After it, we have, after this recognition sequence and cleaving pattern, we have the type of restriction enzyme. So restriction enzyme, so after restriction enzymes are of two types, type 1, type 2 and type 3. After the restriction enzymes, they are of three types, type 1, type 2 and type 3. Now what is type 1? They do not generate specific fragments. They require the presence of magnesium, ATP and another like S-adenyl methionine. The later activates the enzyme. They tracks along the DNA for a variable distance before breaking. These are the three types of restriction enzymes which are there in case of recombinant DNA technology. Then we have type 2. They cut within or immediately adjacent to the target sequence. Generate specific fragments. Then we have the type 3 which requires ATP, which requires energy. Like here type 2 doesn't require any energy but it requires ATP, and so adenylyl methionine for cleavage. Make breaks in the DNA 25 base pair away from the recognition site. So let me show you in the diagram. But these are the restriction enzymes which are there. These are the name of the restriction enzyme like BAMH1, EcoR1, HAE3, HIND2, TFT1. So these are the enzymes, and they have these restriction sites on which they break the DNA molecule. So they have their particular DNA sequence and they have the particular restriction sites on which these restriction enzymes will break the DNA molecule. So if you are able to see here, we have two DNA molecules. This is in white color and this is in black color, and we want to merge these two DNA because in the recombinant DNA technology, the very first step is the joining of two different DNA fragments from different cells or different tissues. So this DNA fragment will be merged with this DNA molecule with the help of an enzyme which is ligase enzyme, but for it we need a sticky end. So we have two types of ends. We have sticky ends when we have the blunt ends. So with the help of the ligase enzyme, we will be able to join these two fragments, and when these two different fragments, if you are able to see, this is totally white, this is totally black. If we are able to join these two fragments, it is known as the recombinant DNA. Now we are able to get the chimeric DNA. Are you getting it? Okay. So are you getting it? So we are able to join two different DNA and we are able to get the recombinant DNA, also known as the chimeric DNA, because the DNA is not the same, but by the use of enzymes and by the use of these restriction enzymes. If you are able to see here, this is EcoR1. So this is EcoR1. This is from the microorganism Escherichia coli. So with the help of this enzyme, we are able to break the DNA molecules, or you can say cut the, not a break, but we are able to cleave that particular DNA molecule, and we get the sticky ends which will bind with the sequence which is complementary to it easily. So that is why we always prefer sticky ends, because in case of sticky ends, the complementary sequences will bind very easily, and what we will get at the end, we will get the recombinant DNA, or you can say we will get the chimeric DNA. So again, this is same thing. If we want to work on RNA, that means instead of DNA we want to work on RNA. So we have to isolate the mRNA first, number one. Then this mRNA gets changes into DNA with the help of reverse transcription. Okay. Now this DNA, it will inserted into the plasmid. The plasmids will inserted into the bacteria, and in the bacteria it will replicate, and we will get as much as we want in our experiments. So this is known as cDNA library. So from the single DNA, when we will get the stock of that particular thing or particular DNA, this is known as the cDNA library. So these are the sticky ends where the complementary sequences like TTAA, here AATT. So the complementary sequences will bind very easily. So these are the sticky ends, and in case of blunt ends, you are not able to get the anything is like any purine or nitrogen base is not here. So the combination or the combining of blunt end is little bit difficult. But kindly do the homework so that you will able to get it. Now, what is the functioning of DNA ligase? I already told you what is the functioning of DNA ligase. It just join the two DNA sequences, the two DNA fragments which are not the same. So ligase will help to bind or help to join that particular DNA fragments. So like these are originally isolated from viruses, also occur in E. coli and eukaryotic cells. So these DNA ligase, the cut DNA fragments are covalently joined together by DNA ligase. So when you are using two different DNA fragments, they are joined together by DNA ligase. This is the enzyme which is quite important in case of recombinant DNA technology. So DNA ligase join the DNA fragment by forming a phosphodiester bond between the phosphate group of 5' carbon of one deoxyribose with the hydroxyl group of 3' carbon of another deoxyribose. So it is they are just letting us know how this ligase enzyme will bind the two opposite or two different DNA sequences. So please try to understand here, the DNA ligase is capable of joining two different DNA together. Getting my point? So this DNA ligase, how it is able, how it is able to join the DNA fragments by forming the phosphodiester bond. So by forming the phosphodiester bond, we are able to join two different DNA sequences together. Okay. So this is the cleavage site, this is a particular sequence. So this is a sequence which we want to isolate. So we just break it with the help of restriction enzyme, and now we have to merge these two DNA sequences with the help of ligase, and when these two sequences will be merged, this is known as a recombinant DNA or chimeric DNA or plasmid chimera. So this is the joining of the DNA. So this is the third step. After it, the fourth step is what? When this DNA will enter into the bacterial cell for further replication. So this is the third step. What is the number one step? Number one step is the isolation. Number two, cutting of the DNA. Number three, joining of the DNA. And number four, amplification of the DNA. So the DNA will be amplified only when this will enter into the plasmid cell. So as we are just start the class, we just So first step is isolation of the DNA. Number two is cutting of the DNA. Number three is joining of different DNAs together. And number 4 is amplification of that chimeric DNA in the bacterial cell. Okay, so these are the 4 steps of the recombinant DNA technology. If you will be able to get it, then you will be able to understand everything regarding our DNA technology. So tomorrow you have to let me know the 4 steps which are there in case of rDNA. So this is your homework. You have to tell me the difference between sticky ends and the main steps or the 4 points in case of rDNA technology. So this is the 4th step where we have to do the amplification. Amplification, or you can say where we need the replica of the gene which we isolated. So what happened here? Now the recombinant DNA, or you can say the chimeric DNA, it is just inserted into the bacterial host strain. So this DNA is get inserted into the bacterial cell so that it will divide again and again, and you will get the many copies of a particular sequence. So these cells are treated with calcium chloride, DNA is added, cells are heat shocked at 42 degrees Celsius, DNA goes into cell by somewhat unknown mechanism. Once in a cell, these cells will, or you can say these chimeric DNA or recombinant DNA will get replicated, and we are able to get as much DNA as we want. So if you are able to see here, we just take the particular sequence, add it in the another sequence, and now this whole plasmid or you can say chimeric DNA enters into the host, and now here it replicates as much as we need. So these are the 4 steps which are there in case of recombinant DNA. John, what happens? Why are you angry? My voice is okay, or there is any noise in between? Please let me know if there is any noise. Is it okay now? My voice is okay? John, is it okay now? Please let me know. Or I will suggest all of you to keep your earphones along with you. My voice is okay, or is there any disturbance right now? Please let me know. Please let me know. My voice is okay now. Hello. Yes, yes, I will share the PPT. Let me complete it, then I will share it. So why, John, why are you so much angry? John, my voice is audible to you. John Weasley. Any one of you, please let me know. John Weasley, please let me know. My voice is audible to you or not? So those who are feeling some background noises, I will request them just keep your earphones along with you. But you are angry right before. You are saying that sounds are like construction worker cutting iron rods for slab. So I think you are frustrated very much. Why you are so much frustrated, John? What's wrong with you? So, John, please let me know the 4 steps which are there in case of RDNA technology. John, please answer the question. The 4 steps which are there in the RDNA technology. Please let me know. Yes, yes. So, John, please let me know the 4 steps which are there in the recombinant DNA technology, please. Isolation, yes, very good, very good. Isolation. Number 2. Yeah, we have to, first of all, we have to isolate the particular DNA. Okay, very good. Next, what we have to do after isolation? What we have to do after isolation? No, no, I am not angry. Please don't take it wrong. I am just asking the question. So, what we have to do after isolation? After isolation, what we have to do? Yes, after isolation, what we have to do? After isolation, what we have to do? Like we have isolated the DNA, then after it we have to do the, any one of you please help him. Yes, yes. So after isolation we have to do the cutting, and after cutting we have to cut with the help of restriction endonuclease, and after cutting we have to join it with the help of DNA ligase, and at the end we have to do the amplification, when we will add that particular chimeric DNA, or you can say recombinant DNA, in the bacterial cell. I hope it is clear to all of you. Okay, is it up to it is okay to all of you? I hope you all are getting it. The only thing is that you will get it. That is the only motive of me. That is why I am asking you the question. So shall we continue further? Shall we continue further? Everything is fine till now. Are you able to get it properly? Please let me know everything is fine. Okay, 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 okay. Thank you all. So after it we have, this is only the basic thing which is there in the RDNA technology. These are the very basic things which are there in the, yes, yes. Soumyadev, you are right. Isolation, then digestion, annealing, and the amplification. So now we will keep moving. Okay, 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 okay. Thank you all. Yes, yes, it's buffering. That is why I just stopped right now. I think it's fine now. Is it fine now? Just wait only. I am not starting it. I'm just waiting. Okay. So I'm just wait all of you, please, please, please. Just wait all of you. So are you getting me now? Everything is no, there's still buffering. So just give me 1 or 2 minutes so that it will be okay, and then we will start the PPT again. Okay, so just wait only. I think it's okay now. No, it's buffering. uh, okay. So okay, it's fine now. I'm just opening the PPT now. I hope you all will get it. Okay. Okay. I'm now just going to start the PPT. So these are the we have basically the rDNA. What is basically here? Now what are the functioning of these enzymes? So there are DNA ligase. I will share this PPT with all of you so you can revise it by your own self also. So we have the enzymes DNA ligase, we have the enzyme restriction endonuclease, reverse transcriptase, polymerase, polynucleotide kinase, exonuclease 3, terminal transferase, and the functioning of each one is given here. So you have to read the enzyme and you have to know the functioning of all these enzymes. Like DNA ligase is basically bind to the DNA molecule. So DNA ligase is basically for the purpose that it will bind to the DNA molecules. Okay, so restriction endonuclease is to for the cleave the DNA at the specific site. So these are the 9 enzymes and I will ask you the functioning of all these enzymes. Are you getting it or is it still buffering? Is it okay now? Okay, okay, okay. Okay. So I'm just showing the present this only this slide that we have 9 enzymes here and you have to tell me the functioning of each enzyme. So this is your third homework. Getting my point? You have to tell me the functioning of blunt end and sticky end. What is the second question which I asked you? This is your third question that you have to let me know about the functioning of all these enzymes. Getting it? So you have to tell me the functioning of all these enzymes. Okay. I will share the PPT right. I will share the PPT. So you have to tell me the function of all these enzymes. Okay. Getting it? So you have to tell me the functioning of all these 9 enzymes. Then after it we have the vectors which are used in rDNA technology. Yes, this is also an important point. So vector is an area of a DNA that can join another part. Like this is one DNA and this is another DNA. So vector is an area of a DNA that can join another DNA part without losing the limit of self-replication. So these all are the vectors: plasmid, lambda phase, cosmid and express sign. These all are the vectors. They are capable of replicating in the host cell. So vectors are basically capable of replicating in the host cell. Should have convenient sites, restriction endonuclease sites for inserting the DNA of interest. Should have a selectable marker to indicate which host cell received rDNA molecule. Should be easy and small to isolate. So these are the vectors. We have plasmid vector, we have cosmid vector, we have lambda phage, we have express sign. So all these vectors we have to discuss in detail. Now what are plasmid vectors basically? Plasmids are small circular DNA molecules that are separate from the rest of the chromosome. So what will happen here? We will use these plasmids, we will insert our own sequence because they replicate independently of the bacterial chromosome. They replicate by their own cells. So we are able to insert the DNA sequence up to 20 kilobase pair. So if we want to work on the DNA sequence whose less than 20 kilobase pair, we can use these plasmid vectors. So if we want to work more than 20 kilobase pair, then these plasmid vector will not work. Everything is fine? Are you getting me? Okay. Okay. So this is the plasmid vector that which is small, which is circular and which is self-replicating when it enters into the instead of the bacterial chromosome. The only thing is that we can insert the DNA sequence of only 20 kilobase pair. If we want to work on the DNA sequence more than 20 kilobase pair, then this plasmid vector will not work. Otherwise, if you want to work on a small DNA sequence, this plasmid vector will work. So these are the plasmids some important points or characters, that they are the extra chromosome, double stranded, circular, self-replicating DNA molecule. Getting my point? They plasmids contribute to about 0.5 to 5% of the total DNA of the bacteria. A few bacteria contain linear plasmids also, because plasmids are generally circular in shape, but we have linear plasmids also. These plasmids carries genes resistance for ampicillin and tetracycline that serve as a marker for the identification of clones carrying the plasmid. So these plasmids have some uh, like tetracycline and ampicillin resistance genes which are present on itself. So, but the only thing you have to keep in mind that we will insert only that sequence of the DNA which is less than 20 kilobase pair in size in this plasmid, which is circular in nature. So this is the plasmid. If we want to insert this purple sequence or this sequence or the foreign sequence, we will insert it inside the plasmid, but only less than 20 kilobase pair. So if you are able to see here, this is the plasmid. With the help of restriction enzyme EcoR1, we are able to get the sticky ends, and this is the purple sequence. If you are able to see, this purple sequence is the interested sequence on which we want to work. So this sequence will get inserted into the plasmid with the help of enzyme DNA ligase. Now this purple sequence should be less than 20 kilobase pair. Now what happened now? This whole plasmid will just inserted into the bacterial cell for further growth. So this is the simple thing in case of plasmid vector. This is the number one vector. After it we have lambda fast vector. Now what happens in case of lambda fast? Lambda fast vectors are recombinant infections containing the phase chromosome and in addition to the embedded outside DNA. All in all, fast vectors can convey bigger DNA grouping than the plasmid vector. So fast vectors basically they will take the DNA most the size of which DNA will be more than 20 kilobase pair. So they are the vector. Now another is cosmids. So cosmids are the vectors possessing the characters of both plasmid and lambda phase. These carry the larger fragments of foreign DNA compared to plasmid. So cosmid are another which will carry the DNA fragments up to 45 kilobase pair. So if you want to work on long sequences of the DNA, then you have to go with cosmid vectors. So they can replicate like the plasmid but can be packed like the phase lambda. The cosmid vectors have the properties of both plasmid vectors as well as the properties of the lambda phase also. So we can also go with cosmic vectors if the size of the DNA fragment is more than 45 kilobase pair. Then we have another vectors which is expression vector. This is also used to carry the DNA of interest. Then we have bacterial artificial chromosomes. In this we are able to get the DNA of 300 kilobase pair. I am not going in detail regarding these vectors, but you have to keep in mind that there are different vectors which will carry different sequences of the DNA. According like this will carry 30 kilobase pair of DNA. So you have to keep these things in mind only. After it we have the techniques which are used in rDNA technology. In the rDNA we are using gel electrophoresis, we are using cloning libraries, we are using restriction enzyme mapping, PCR, nucleic acid hybridization and microarrays. All these techniques will be discussed in this lecture series in detail, so I don't think I will cover it here. So these are some applications of DNA, rDNA technology, like I already discussed it before also, that when we want to work on a specific disease, when we want to diagnose some diseases like sickle cell anemia, thalassemia or cystic fibrosis, when we want to work on, you know, group of diseases, or we want to diagnose some molecular diseases, for that purpose rDNA technology is needed. Even it is useful in gene therapy, it is useful in agriculture also. So you can go and you can have to, you know, This gel electrophoresis I will discuss it in detail, not here, but in the another lecture series. All these techniques will be discussed later also. So that is only in case of rDNA technology. Okay, the techniques which are there, the gel electrophoresis, the PCR, then your nucleic acid hybridization, I will discuss those techniques in another lecture series. Up to now, up to it, is it clear to all of you? Because I just give you the only the basics which are there in the rDNA technology. I am not taking it as, you know, I am not taking much deeply regarding this, but at least you should know what is rDNA technology, what are the main steps which are there in the rDNA technology, and how we have to, process it, how to understand it in detail only. So this is the thing which I want to do in rDNA technology. Then after it we have some applications of rDNA technology, the same applications which are written on in your PPT which I just show you before. So these are some of the applications, like it is useful in the production of vaccines, it is useful to cure some diseases, it is useful to make, like this is HB vaccine, when we want to make some vaccines related to a particular disease. It is useful in pharmaceutical industries also. Okay, when we want to generate some growth hormones, all for all those purposes, these rDNA technology is needed. For DNA fingerprinting, for gene therapy, for molecular diagnostic of the disease, these therapies are used. Even for this HIV/AIDS, this technology is used. We are using these technologies, and these is also used in the field of agriculture. So I will share this PPT. You have to read these by your own self, and if you will not able to understand anything regarding the applications, you can share it with me, and I will let you know regarding it. But first just go through that the PPT of this and the PDF of the applications of the rDNA technology. If you will not be able to get it, then definitely I will help you. So is it okay to all of you? I will share this thing right after your lecture. I will share the PPT and I will share the PDF in which we have the applications of the rDNA technology. Now I have to move further to another topic, which is blotting and which is PCR. I think we will go with PCR. So is it okay for today? Is it fine, or we will start the another topic also? Please let me know, because I want to start PCR also. But if you are feeling, if you feel bored, then no issues at all. We will discuss PCR tomorrow also. But let me know so that I will. If you have some time, if you have some, you want to, then I will start the PCR also. PCR is basically polymerase chain reaction. I hope many of you have done it before also. Please let me know if you have done PCR. You must be know about PCR because it is there in your syllabus, in your bachelor classes also. So is it fine that we will start PCR? Have you done it before also? Any one of you, please let me know. Okay, okay. So have any one of you have done PCR before also? Any one of you have some knowledge regarding the PCR? Please let me know. Because the signal is fine today, that is why I am just feeling that let me complete PCR. Okay, I will start it right now, but just let me know, have you done PCR before also? Is there anyone who is new to PCR? If anyone is new to PCR, then I have to start from very basic. Otherwise, I will do it little fast also. Anyone? Don't be shy if you have not done it before. No issues at all. Just let me know if you have not done it. No issues at all. I will discuss everything. Just let me know whether you have done it or not done it. Okay, okay. So you must have done it before also. And yes, someone has worked on RT-PCR also. Very good. So John is currently working in a lab with RT-PCR machines. Very good, very good. So you must have studied it in the, you must have studied it in the in your bachelor classes for your theory exams definitely, because lots of teachers are there who will definitely ask some questions from the PCR or the basic steps which are there in the PCR. Okay, 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 okay. I will start. I will start. Okay. So I hope you all know it, but sorry if you feel it very basic, because I have done uh, related to the students which are very basic to it. So I will start it very fast. And okay, 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 Murita. Definitely I will start PCR in very, but I will try to do as much as I can. Okay, so I am now just going to start it. So yes, we have another term which is, oh sorry, we have to discuss a very simple molecular biology technique which is known as polymerase chain reaction. Now what is basically polymerase chain reaction? Polymerase chain reaction is a technique which is used in the molecular biology to amplify. Yes, now I will ask you one question after this PCR. That will be my last question, and that will be your homework also. So you have to complete those 4 questions which I asked you today, and you have to write it in your copy, click the photo, and you have to put it in a telegram group, and I will check it very deep, very thoroughly. Okay, so at the end of this PCR I will ask you the last question, which will be my last question of the day. Okay, so polymerase chain reaction is a technique which is used in molecular biology to amplify a single copy or a few copies of a segments of a DNA across several orders of magnitude, generating thousands to millions of a copies of a particular DNA sequence. So with the help of PCR, what basically we are doing, we are able to amplify a particular DNA sequence. We are able to get the thousands or millions of copies of a particular DNA sequence. It was developed in 1983 by Carey Mullis. In 1993, Mullis was awarded the Nobel Prize for his work on PCR. So what are the components which are there in the PCR? In the PCR, the very basic thing which we need is the DNA template, the DNA segment which we want to amplify. Getting my point? Suppose we want, this is the whole DNA, but only this segment we want to amplify. So the segments were concentrated, which is the target sequence. Number 2 is the DNA polymerase. Now this is the enzyme which is needed in the PCR. So DNA polymerase sequentially adds nucleotides complementary to the template strand at 3' OH of the bond primers and synthesize new strands of the DNA complementary to the target sequence. The most commonly used DNA polymerase is tag DNA polymerase. But now there are some another enzymes also which are used as a DNA polymerase. Like we have PUFU DNA polymerase from Pyrococcus viraceus. Along with it, we also need magnesium ions as a cofactor for DNA polymerase. So for the PCR reaction, we also need magnesium ion as a cofactor. Primers are needed. Now what are the primers? Primers are the synthetic DNA strands of about 18 to 25 nucleotides complementary to the 3' end of the template strand. DNA polymerase starts synthesizing new DNA from the 3' end of the primer. So these are the primers. Primer designing is there, which is you must know about it, that which type of sequence on which we want to work. Then we need DNTPs, also known as deoxynucleotide triphosphates, which are the building blocks for the synthesis of new DNA strands. We need magnesium ions, as I told you before also. Now this is the procedure. We have to do denaturation at 94 degrees Celsius. So the double stranded will become denatured at 94 degrees Celsius. After it, we have to do the annealing. After annealing, we have to do that, like this is annealing at 54 degrees Celsius. We have to run the primers. We have forward primer and we have the reverse primer. So we have to run it, and after it we have the extension at 72 degrees Celsius, when we are able to get the replica of the original or the parent sequences. So we will able to get the replica of that, or we are able to amplify that particular DNA sequence. I will share the PPT. You can go it by your own self also. So this is the template DNA. By first cycle we will get the 4 copies, but by the 35th cycle we are able to get the 60 billion copies. So by 2 hour or 2.5 hour processing of the PCR, we are able to get the that much amplification of a particular DNA segment which we want to work on. So these are the general protocol which is there in case of PCR. We need water, we need magnesium, we need DNTPs, we need forward primer, reverse primer, tag polymerase, and the template DNA. So these all are the things when you are working on a PCR basically. This is the PCR machine, and these are the applications of the PCR. I will share the PPT and you can go it by your own self. So I hope it is little bit fast, but I'm sorry, but I know you all know it, so that is why I'm not putting much, you can say much work on this one. I know you all know it. I will share the PPT with all of you so that you will read it by your own self. Yes, Rubin, RT-PCR is not just like the PCR. The simple PCR is very basic, but RT-PCR is little bit advanced. Okay, so there are some modifications there, and there are some graphical representations also in case of RT-PCR when we are working on the real-time PCR. So I'm just tell you only the basics of PCR. I'm sorry, but I know you all know about it. So that's all about the PCR and about the RDNA technology. I will share the PPT right after this lecture so that you can go by your own self. Okay. Now I have one question which I want to ask you, but you have not to reply me right here. Any one of you please write the 3 questions which is your homework. Then I will ask you the last question of today. Any one of you please share the 3 questions which we have to do. Yes, yes. So you have to let me know about the difference between sticky and the blunt end. The third question is about the 9 enzymes, the functioning of all those enzymes. Now, what is the second question? I also forget. What is the second question which I ask you to do as a homework? What is the second question? Any one of you remind? Any one of you are able to remind? Because yes, this is the first question, and the third question is the 9 enzymes. But what is the second question? I also forget. Any one of you have you anything keep in mind? Yes, yes, Shreya, I will let you know about RT-PCR also, but for it I have to take the photographs of RT-PCR so that we will able to share everything in detail. Okay, okay. Yes, the second question is the steps of RDNA technology. Okay, 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 okay. Getting it, getting it. So the very first question is, what is the difference between blunt end and sticky ends? Number one. Number two, the steps which are there in the RDNA technology. Number three, the enzymes and the functioning of that particular enzymes. And now the last question, which is the fourth question, and you need not to have to write here, because I want you all to think about it, and you have to let me know in the telegram only by writing it in your copies. Now just let me ask you the fourth question, which is quite important. Yeah. In case of recombinant DNA technology, in case of rDNA technology, we are taking the DNA sequence, we are merging it with another DNA sequence. Then what we are doing? What we are doing? We are taking that chimeric DNA into the plasmid, into the bacterial cell, and then in the bacterial cell we are amplifying that particular segment, right? We are amplifying that particular segment. Is it right? So in the case of RDN, this thing is that we are amplifying a particular gene, suppose insulin gene, with the help of bacterial cells. Similarly, in the case of PCR, we are doing the same thing. We are just isolating the DNA. We are just taking the particular DNA sequence, and after that particular DNA sequence, we by using 2 hours long machine, we are able to amplify that particular DNA segment. So if PCR is quite enough to get us thousand to billions of copies within 1 hour or within 2 hours, what is the need of RT-PCR technology then? I repeat my question again. When we are able to get the sequence, we are able to amplify the DNA sequence with the help of PCR. Getting my point? We are able to amplify the sequence with the help of PCR. We get thousands of sequences by 2 hours only. So what is the need of doing those steps which are there in the rDNA? We have to cut those sequences, then we have to merge it with the plasmids, then plasmids will enter into the bacterial cell, and then the bacterial cell will replicate. So this is my question. You have not to answer here. You have to, are you understand the question or not? I am asking, when PCR is there, PCR is for the amplification of the particular DNA segment, then why there is a need of rDNA technology? This is my question. I hope you all will get it, and it is okay for the day. You have to answer, you have to think about it, and you have to let it me know by tomorrow only. So you have to answer those 4 questions in your copy, click the photo, and you have to paste it in a telegram. No, you have to paste it in your telegram group. All of you have to paste it in a telegram group. Okay. So these are the 4 questions which are your homework. Uh, John, I already. Okay, let me ask you the question again. Like PCR is here where we are able to isolate, or amplify the particular DNA sequence, right? Within 2 hours, within 3 hours, we are able to isolate particular DNA sequence. So when we are able to do it with the help of a PCR, why there is a need to do it with the help of rDNA technology? Why we have to isolate the sequence, then we have to immerse it in the plasmid, then we have to enter it in the bacterial cell, then we have to do the replication. So why there is the need of the rDNA technology? I will not write the question is only this: when there is a PCR for amplification, why do we need rDNA technology? PCR is there for amplification, then why do we need rDNA technology for amplification? That's it. This is the question only. I think you all get it. If any one of you understand the question, please type it in the telegram group also. I will request any of you and even all of you to write all the 4 questions on the telegram, so that all will be able to answer the questions. Okay. So yes, Yamin, I ask you that don't answer here. I just want the answer tomorrow, not right now. So that's all from my side. So it's the long lectures today. I think that's all for today. We will meet tomorrow. Till then, take care of yourself. Good night from my side. Bye bye. If you want to ask anything, then you have 5 to 10 minutes more. Otherwise, we will meet tomorrow. So please my request to all of you to please write the questions in the group also, so that all will understand it. Okay, okay, okay. So thank you. I just want to thank you all for your patience and for your support, which is there. Yes, yes. So I just want to say thank you to all for your continuous patience along with me. You are continuously here along with me for such a long time. Thank you all. Thank you all. We will meet tomorrow. Till then, take care of yourself. If you have any, oh, Jeet, how are you? Why you are here right now? Okay, okay. So Jeet is my previous student from my another lecture series, and they are very cooperative and very good students of mine. Yes, yes, I miss you all. Hope you all are fine and doing great in your exams. Everything is fine in your side? Yes, yes. Bye bye, Subsina. No, Narita, I will share the PDF with all of you. There are 9 enzymes which are basically you have to let me know the functioning of every enzyme. Yes, yes, Jeet, you are always welcome. You are always welcome from my side. And when you will be free, then please join the lecture series and you will get the knowledge as much as I can share. So you all are welcome. So yes, thank you for being here. It's so nice that you are here for messaging me today. Thank you for being here. So Cheta, you are so much focused on the timing. I will let you know about the timings. Don't worry. We will take it on the tomorrow is Monday. We will take it on the scheduled time. But yes, I already told you 2 3 days back that on Saturdays and Sundays on weekends I will take little bit long action, long classes. Sorry. So yes, please don't worry about the timings. Please, please. So that's all for today. Please take care of yourself. And Jeet, please be here when you will be free. It will be nice if you are here. So that's all for today. Take care of yourself. Good night. Good night. Take care of yourself. All of you, please. Okay, take care. Take care, all of you. Please take care. Good night. So yes, take care of yourself. Now I am just going offline, and as I have to upload the videos also in the group. So that's all from my side. Bye bye. Take care all. Bye bye. No issues, Dipti. Just try to get it theoretically also. So when you will get the opportunity of using the labs, then you can also do it practically also. No issues at all. So take care of yourself, please. So bye bye all. Bye bye. Please take care of yourself. Good night. Bye bye. So I'm now just going to upload the videos. Sorry, not the videos, but the PPTs in the group. So bye bye. Bye bye from my side.